Hello. We'll spend the next 5 lectures discussing nicotine addiction. It's the only addiction that we will discuss in detail in this course. There are several reasons for this. First, it's the only addiction that I can describe with great familiarity. I've worked on basic mechanisms of nicotine addiction since around the year 2000. And I think that we can drill down to some important molecular mechanisms. It is certainly the most widespread addiction in the modern world. More than 1 billion people use smoked or smoke-cured tobacco. And just as importantly, nicotine and the nicotinic system and nicotinic receptors have really served as a touchstone, a point of departure for neuroscience. Ever since Columbus's crew sampled tobacco in 1492. And so we have a lot to learn in neuroscience in general from studies on nicotine and nicotine addiction. Let's talk about the components of nicotine dependence. Nicotine dependence, and we will use the term somewhat interchangeably with addiction. Nicotine dependence has several components, probably more components than opioid or cocaine addiction. And yet people are able to function very well when they take nicotine, whereas when they take opioids, they are often drowsy, and when they take cocaine, their judgment is often clouded. And this is why so many people are dependent on nicotine. The nicotinic receptors occur in many brain areas, and actions in these brain areas probably control each component of nicotine addiction. So there is reward. People get a feeling of well-being when they use nicotine. That is probably in an area that I will tell you about in the next lecture, but in brief, you remember the handlebar mustache, and you remember that the upper lip of the handlebar mustache is involved with a sense of well-being, the ventral tegmental area. Cognitive sensitization. Many people say that they think better when they smoke, or that they learn better when they smoke. And it's impossible to ask a person whether he thinks or learns better when he smokes than if he did not start smoking in the first place. It's a bit more straightforward to do this with animals, and it is very clear that animals who have been given nicotine for a week or two and then withdrawn from nicotine do not perform well on cognitive tasks. But if they are then given another dose of nicotine, they perform much better. Now, do they perform as well as they would if they had never been given nicotine at all? This question can be asked, and the general answer is no. Even after nicotine chronic application and then a bolus of nicotine, animals' cognitive performance is generally not so good as it would be if they had never been given nicotine. Cognitive sensitization probably occurs in the forebrain, in the cerebral cortex, or in the hippocampus. Other people smoke because of stress relief with nicotine. Not clear where this occurs, probably several brain areas. Other people continue to smoke because they anticipate that when they stop smoking, they will gain 5 or so kilograms. And this is a common occurrence when people stop smoking. Some people smoke for a kind of self-medication, especially in schizophrenia. We'll discuss this in a later lecture. And a large number of people take nicotine to avoid withdrawal symptoms. Let's make the general statement that nicotine-dependent people value the effects produced by the smoking-induced nicotine pulse or bolus, and that this bolus either activates and then desensitizes nicotinic receptors. But longer-term exposure is essential for nicotine dependence. The meaning of longer term depends on one's definition of nicotine dependence, at what point in life one started smoking, but the time required may be as brief as a few days, although we usually think that it takes weeks to develop nicotine dependence. And therefore, most of the data that I'm going to review for you over the next few lectures, and most of my own research, has been involved with asking the question, what changes in the brain during chronic exposure to nicotine? Chronic does not have the same kinds of connotations during this use as it does for chronic disease, although drug dependence is called a chronic disease. For chronic, we actually mean repeated or continued exposure to nicotine. Now, understanding the events that occur in the brain during chronic exposure to nicotine has to be accomplished at a large number of levels. We need to think about, starting at the top, we need to think about behavior, about circuits that govern behavior, about synapses that control circuits, about neurons that contribute to these synapses, the intracellular events that change during chronic nicotine, binding to receptors, the differences between nicotine and the endogenous transmitter acetylcholine, the proteins involved, the RNAs involved, and finally the genes involved. Many of these do change during nicotine addiction. And as you can by now appreciate, I'm interested in the most fundamental biophysical aspects of these changes. But let's begin at the top. During these lectures, we're going to begin at the top and work down to the most basic mechanisms. And a good place to start is the Fagerstrom test for nicotine dependence, which has been used in its present form for more than 20 years. It's modified from an earlier test by Fagerstrom for tolerance. And this test does emphasize withdrawal and tolerance. And it de-emphasizes such aspects as cognitive sensitization. And perhaps it also de-emphasizes stress relief. So there are a series of questions, 6 of them. And so when your friend asks you, am I addicted to or dependent on nicotine, you can give your friend the Fagerstrom test and add up the points and see where your friend falls on this scale. How do we ask animals whether they derive reward from nicotine? A classic and non-invasive test is called conditioned place preference. And in this test, the animal is given an injection of nicotine in an environment that he can recognize. And he is then tested in 2 chambers. One is the chamber where he received the drug. The other is a neutral chamber where he did not receive the drug. And he can decide which chamber to enter. And so this tests whether an animal feels reward associated with the chamber where he got the nicotine. Now, there are several controls that need to be done. For instance, you have to make sure that the animal is not attracted to or repelled by the handling and by the injection itself. And so there are lots of controls with saline. You have to make sure that the animal is not remembering other animals in the chamber. The experiment can be expanded so that the animal can receive different injections and different drugs in the 2 chambers. And so you can ask how much the animal values one drug over another. So that is conditioned place preference. Probably a test that seems to be more generally applicable to human behavior is one where the animal is asked to perform a behavior to get some nicotine. Now, this is called nicotine self-administration, And it is done quite reliably and robustly with rats. It is a much less reliable experiment with mice. These experiments do not go on in my lab, and so I'm telling you what my friends tell me. So the animal is taught to press a lever which will give him either nicotine or the control solution, saline. It takes a few days for the animal to get accustomed to this, and he eventually does it very well. And then you ask the question, what dose of nicotine elicits the best response? And a very common phenomenon in self-administration experiments is the so-called U-shaped dose-response relation. An animal will press a lever very reliably for increasing doses of nicotine, but after a while, as the dose increases more, the animal seems to dislike this, and he presses the lever less often. So this needs to be explained, and it is explained rather simply in terms of the combination of a reward pathway and an aversive pathway. At low doses, the animal is getting a reward, and at higher doses, he seems to be averting some unpleasurable stimulus. So we have done dose-response relations here, and we can model this. This is not a real experiment, it's a model experiment in which the rewarding pathway has a lower EC50 than the aversive pathway. We'll say their efficacies are the same and their Hill coefficients are the same. And to get aversion, we simply take the usual dose-response relation and we subtract it from 1. And the spreadsheet, if you would like to manipulate this yourself, is on the website. So when we take these two dose-response relations and multiply them together, the rewarding pathway and the aversive pathway, we get this U-shaped dose-response relation. So this is a formal way of saying, very simply, the animal likes lower concentrations, dislikes higher concentrations. Where is this taking place in the brain? Well, there's some indication that the rewarding pathway is centered on the ventral tegmental area, and recent studies suggest that the aversive pathway may involve a smaller area called the medial habenula. Another point about chronic nicotine is that we can measure tolerance to dopamine release. In order to measure tolerance to dopamine release, experimenters use two animals. They use a so-called master animal that has been taught to self-administer either nicotine, and the control, as usual, is saline. And we want to ask whether this changes a measure of reward in the brain. So we're going to be measuring dopamine release. To do this most fairly, the experimenters don't use the animal that's been trained, but they use another animal, the so-called yoked animal. The yoked animal gets pulses of nicotine at the same time that the master animal does, but the yoked animal is not doing anything and therefore does not know that he's going to get the reward and cannot use dopamine to anticipate the reward. And it's the yoked animal which has a tube, a cannula, and so the analysis then gets done on the question, how much does the yoked animal secrete dopamine in this case, in the nucleus accumbens, I believe. And so if the animal is yoked to saline, that is, not getting nicotine injections, but only saline injections, the animal has a certain level of dopamine secretion, and then when tested by another nicotine dose, increases the output of dopamine. So this is comprehensible and easy to understand. Now, what happens if the test animal is yoked to doses of nicotine instead? Well, the animal has a lower baseline level of dopamine release, and this could be equated to tolerance, and it could be equated to the animal wanting more dopamine. And furthermore, a test nicotine dose produces a blunted response, less dopamine. And this could also be part of the tolerance process. So here is a real pharmacological, biochemical correlate of tolerance in a well-controlled animal experiment. Next time, we'll talk about circuits and cells.